Okay, so hello everyone. This is group one. We will be discussing about antibiotics, and our team includes Ayang, Bomi, Jiyoon, Uichan, Minsung, and Hugh. We will be discussing over four sections, which is definition, mechanism, application, and limitation of antibiotics.
Okay, the definition is as written in the slide. Antibiotics are medications used to kill or destroy harmful bacteria that's embedded into human body, and many people misconcept that uh, the antibiotics and the antibiotics are exactly the same things. However, there are certain distinctions between the antibiotics and the antibodies. The antibiotics, as the written in the slide, it usually kills the harmful bacteria, while the antibodies are used to increase the immunity of the human body.
So mechanisms and inhibit cell synthesis, and some antibiotics prevent bacterial cell synthesis and eventually causing bacteria to live by osmotic pressure. And examples of major antibiotics that inhibit bacterial cell synthesis include penicillin and cephalosporin, which are also called beta-lactam because of the four-member ring common to all members.
Another possible mechanism used by antibiotics is inhibiting protein synthesis. Bacterial ribosomes, each made of a 30S and 50S subunit, synthesize proteins through a process called translation. Some antibiotics target this specific process to prevent protein synthesis, and how this works depends on the type of antibiotic. For instance, aminoglycosides bind to the 30S subunit to cause misreading and premature termination of translation. Tetracyclines block tRNA from binding to the A site of ribosomes, preventing a necessary part of translation. And macrolides bind to the 50S subunit and prevent elongation of the peptide chain. The third mechanism is inhibiting DNA replication. Some antibiotics prevent bacteria's DNA replication, making bacteria unable to multiply. For instance, fluoroquinolones inhibit bacterial enzymes necessary for DNA replication, such as DNA gyrase and topoisomerase 4. These enzymes are necessary in dealing with DNA supercoiling during replication. And the fourth mechanism is inhibiting folic acid synthesis. These antibiotics inhibit bacteria from synthesizing folic acid, which is crucial for growth and multiplication. These antibiotics target specific enzymes involved in folic acid metabolism. And sulfonamide and trimethoprim are examples of antibiotics that target specific enzymes that work in the folic acid metabolism pathway.
Now let's move on to application of antibiotics. The first example of antibiotics is penicillin. Penicillin is the first antibiotic discovered by Scottish bacteriologist Alexander Fleming in 1928. Fleming serendipitously found areas around an invading fungus in a petri dish that were immune to bacteria mold belong to a Penicillium genus. Penicillin uses the mechanism of inhibiting cell wall synthesis, which is already explained in the previous slide. Penicillin binds to molecule on the walls of bacteria and prevents the target from forming the walls when actively multiplying. The target bacteria eventually gets ruptured. Penicillin is effective against gram-positive pathogens. And let me briefly explain what gram-positive and gram-negative bacteria are. So gram-positive bacteria are the ones with thick peptide double layer, which prevents the cell from bursting. and the gram-negative bacteria have thin peptide dog-lichen layer, but they also have other layers that block antibiotics attacks. And penicillin has an antibacterial effect on H. influenzae, E. coli, Salmonella, Shigella, and Pseudomonas. It can also be used to treat pneumonia, blood infections, uncomplicated gonorrhea, meningitis, and endocarditis.
The second example of antibiotics is cephalosporin. Cephalosporin is a broad-spectrum bacteria first discovered in 1945. Though cephalosporins are not usually used as a first-choice antibiotic, they are used when other antibiotics cannot be used. Normally, cephalosporins are used for patients in whom the eradication of the infection is critical. Cephalosporins kill gram-positive bacteria by binding and blocking the activity of enzymes responsible for making peptidoglycan, which is an important component of the bacterial cell wall. Just as penicillin introduced earlier, cephalosporins also inhibit bacterial cell wall synthesis. By inhibiting cell wall synthesis, the growth and multiplication of cells would slow down, resulting in the interference of bactericidal action. Lastly, cephalosporins are used for bone, ear, skin, upper respiratory tract, or urinary tract infections.
The third example of antibiotics is Quinolone, which is a broad-spectrum antibiotic discovered in the early 1960s. Quinolones work by inhibiting DNA synthesis of both gram-positive, such as S. aureus, and gram-negative bacteria like Haemophilus influenza by primarily targeting two enzymes, DNA gyrase and topoisomerase IV. DNA gyrase is a crucial bacterial enzyme that triggers the directional supercoiling of DNA, and this process is essential for chromosome condensation and RNA transcription. Since this particular enzyme is only present in bacteria and indispensable for bacterial growth, it is the best target for quinolones.
Quinolones are used to treat bone infection, bronchitis, cholera, E. coli infection, pneumonia, and more types of bacterial infection. Despite the wide usage of quinolones, there is a critical issue caused by their overuse. Currently, the number of quinolone-resistant bacteria is rapidly increasing, and therefore the effectiveness of antibiotics in quinolone classes is constantly decreasing.
And we'll now be discussing about the limitations of antibiotics. We'll go over two major limitations. First one is antibiotic resistance, and the second one is side effects. So antibiotic resistance, it's a pretty much self-explanatory name, but antibiotic resistance is when germs like bacteria and fungi develop the ability to defeat the drugs designed to kill them. So this can typically happen via mutation in bacteria structure, and two keywords are directional selection and plasmid. Directional selection is the keyword because antibiotic resistance is a type of directional selection, and usually bacteria contain antibiotic resistance gene inside the plasmid, which is a short strand of DNA with specialized functions. So how does antibiotic resistance work? So for this, we have specific example, which is penicillin. So usually beta-lactam structure of penicillin inhibits formation of peptidoglycan cross-link in bacterial cell wall. But however, when mutation arises in gene that codes for the bacterial cell wall, this disfigures the cell wall of the bacteria, which results in a cell wall that cannot be cut by the penicillin. So therefore, since the protein shape has changed, penicillin cannot bind to the active site anymore, which results in non-functioning antibiotic and a bacteria with a mutation that is no longer susceptible to penicillin. So bacteria in the colony with this special trait will be able to survive and thrive and reproduce, which results in a resistant offsprings, which is a type of directional evolution. So we have this small diagram about how antibiotic apocalypse is currently happening. So the example we have here is again penicillin, and the bacteria we'll be specifically looking for is Staphylococcus aureus. This is a bacteria which typically causes skin irritation if it's contacted via the skins, or if it's ingested and orally, it can result in vomiting, headaches, or tummy aches. So usually the treatment for this bacteria was to apply penicillin. However, as I mentioned before in the previous slides, now the antibiotic resistant genes are now passed down to the next generations of this bacteria, which results in antibiotic resistant bacteria to thrive within the population. And then the scientists had a treatment to this. However, the bacteria soon developed antibiotic resistance to this treatment, and now it's an ongoing chain, and at the end we might run out of antibiotics and we might not be able to have solutions to bacterial infections anymore. So what is the potential solution? So currently, over-medication is the major factor contributing to the resistance bacteria's prosperity. So as you see in the GIF right next, 30% of antibiotics are prescribed unnecessarily. So patients with cold is prescribed with antibiotics to prepare themselves for secondary infection, which is a over-preparation. And according to World Health Organization, they recommend us to only use antibiotics when it's prescribed by a certified health professional and never demand antibiotics from health worker.
Not only the emergency of the antibiotics is the problem, the medication itself, it has the side effects, which makes the certain limitations on the antibiotics. The first side effects is you can expect from the antibiotics is certainly allergic reactions. And you might think that I usually take the antibiotics often, but I don't have the allergic reaction. But certainly, there's a lot of cases for the allergic reactions in the world. And the second one, as I mentioned before, as a definition of the antibiotics, it kills the harm bacteria, but it does not only kill the bad bacteria, it also kills the good bacteria, so that it makes human cells sick of it. And for the third one is if the pregnant woman dose the antibiotics, then it is extremely detrimental for them, and etc. Skin rash and nausea and a lot of the side effects on here. Thank you. This is the end of our slides. These are our source. So far, this was the Group 1 H2O Club. Thank you.